Good morning, everyone. My name is Dmitry Petrovic. I am from the International Nanotechnology Laboratory, one of the partners in the INFRACHIP project. As you may remember from registering for this series of webinars about sensors, it is brought to you by the partners of the INFRACHIP project. The webinar today is going to be about the sensors for sustainable agriculture and the environment. Our speaker is a head of the Precision Electrochemical Nanosensor Group at the Tyndall National Institute. In particular, the group is developing two complementary sensor technologies on silicon chip substrates that employ nanoelectrochemistry and surface-enhanced Raman spectroscopy detection mechanisms. We're going to hear, obviously, in a bit more detail about both of these. And in particular, the group is focusing on trying to ensure that the technologies being developed are relevant in real life deployment environments. And so a lot of this work is done in kind of collaborative co-development. And again, we're going to see some examples of this shortly. I would like, again, to point out, and we will remind you about this again towards the end, that the idea is that these platforms, these sensor platforms that you're going to hear about today, in principle, access to them or something similar would be available through the INFRACHIP project. If you see something that you are interested in, something that you like, then definitely feel free to get in touch with the INFRACHIP project, and we will try to see what we can do. And basically, we're going to go ahead and start the presentation. There will be a question and answer period towards the end, and I encourage you to use the question and answer facility that you can find. Well, I guess typically it's at the bottom of the screen, but basically there is the question and answer tab where you can write the questions. You can write the questions during the presentation or at that final period when we're going to get to that in the end. And with this, I will let our speaker begin the presentation.
So thank you very much, Dimitri. Good morning, everybody. My name is Alan O'Riordan. I'm from the Tyndall National Institute in Cork, in Ireland. I lead a precision nano electrochemical sensors group, most of which I'm going to talk about this morning. I won't go much into the Raman spectroscopy, but what you'll see this morning is also equivalent in Raman. What I want to talk about today is Tyndall is obviously a core partner in INFRACHIP. We have our own silicon microfabrication, nanofabrication facility at Tyndall. That's our unique selling point. It's the only research fab on the whole island of Ireland, north-south. We're based in Cork, down the very south of the country. We have multiple clean rooms. We have multiple characterization suites. We have we started in microelectronics, the National Microelectronics Research Centre, but we have an awful lot of expertise now in photonics, in chemistry, and life sciences, which we bring together in a multidisciplinary, kind of interdisciplinary environment at this point. And we collaborate widely across Europe in EU framework programs. We've national funding, we've US Ireland funding as well. So we're very active from a funding point of view. What I would like to talk about this morning is the nanoelectrochemistry element, the work we're undertaking in my group. But rather than going into a specific technical presentation on the things we've done and the different papers we've had, what I've tried to do is to break this out and to kind of put it into a context where other people who might be interested could maybe use some of the technologies or approaches that we have in developing our sensors and access them through INFRACHIP. So, for example, if people were doing sensor design and they wanted some simulations done, well, we could look at doing that, or materials if you want to do some material synthesis or deposition for selectivity, we could also help it with that. So that's the way I've broken out my talk this morning. Obviously, there's an awful lot of in-depth stuff behind that. I'm very happy to take questions and answers at the end of this talk. So without that, I'll start. So here, if you can see my my mouse, there we go. So this is Tyndall. Like I said, we're located in Cork. The sky here is airbrushed in this blue sky. Normally it's raining, so we just pretended. So we put in some blue. No, not really. This is the one sunny day that we had last year. So we've shiny new laboratory buildings. We're situated in an old maltings facility here. This building on the right is over 200 years old. It's a listed building. It's now filled with labs. And you can see some of the examples of some of the work we're doing below. Okay, so Tyndall itself is broken into two main research centers. We have the Micro Nano Electronics Systems Center where I sit, and we also have a Photonics Research Center, but basically focused on telecommunications, biophotonics, that kind of thing. Like I mentioned earlier on, we're underpinned by this specialty products and services, which is our research fab and characterization type of a suite of analytical instruments. We focus in four main areas. So we're looking at agri-tech, food and the environment, which I lead at the moment. We're looking at health and life sciences, micro and macro energy, everything from wind turbines down to energy on chip and energy harvesting, and ICT, information communication technologies. My space, we're playing in agri-food. You might ask, why is there a kind of microelectronics institute looking at agri-food? And simply the answer is very simple. The global population is growing. It's expected to hit 10 billion by 2050. We have to feed everybody on the planet. We have very limited resources. We have to make sure when we are growing these materials or these food, that we do it in a very sustainable way. And there's a number of different directives from EU and from the UN that are driving sustainable food development to reduce wastes, reduce losses, and increase productivity. And of course, to do what we have to do all that against the backdrop of climate change, the competition for land. So there's going to be 2 billion extra people. They're going to have houses. So if we build the houses, we can't grow food in the land, and a lack of biodiversity. So the only way to address this challenge really is to digitalize the agri-food environment. It's a very traditional, as you imagine, agricultural environment where it's passed from family to family, and the work practices that are carried out are basically, a lot of them are done because they've always been done that way. So there's a need to now develop a whole suite of IoT tools that will enable the farmer and the producer to comply with different directives such as nitrate losses or greenhouse gas production, because at the moment there is no real tools available, digital tools available to the end users. So in that context, that is the work that we're doing in Tyndall. And to do that, we set up what we call the Sustainable Agri-Food and the Environment Research Cluster, Strategic Research Cluster. And in that, we have multiple PIs, different groups, all working to develop hardware solutions that will provide farmers and stakeholders with real-time decision-making capacity. So basically to allow them to be do the right thing at the right place at the right time with the right amount. So it's key that these are kind of rapid type of tests that give the farmer mostly and maybe producers or regulators key information that's important to them. So what we have what we do at Tyndall is we work across a wide range of TRLs. We work at a very, very low TRL level. So we undertake different types of COMSOL simulations, electric field, diffusion, transport. And these simulations give us an indicative knowledge on how a sensor should look, what the architecture of it should be, if you're doing, say, the micro electrodes or nano electrodes for sensing, the layout of those electrodes, what the dimensions of them should be, the gap, the separations, etc. So these simulation tools then can, once they're developed, once the different models are developed, can be applied across a very wide variety of different types of sensor designs and architectures. So once we have information on how they should look and how they would behave, we then will go away and use the fab, and we'll fabricate them on site. So for the we have a full optical photolithography suite, and we also use electron beam lithography, and we have a nice hybrid method that allows us to swap between the two of them. So we use the e-beam for the nanoscale stuff, and then we use the optical lithography for the interconnect and all of that. And we develop a variety of different platform sensors. So you'll see in the presentation as I move on, there'll be some architectures there that we use for chemical sensing, for biochemical sensing, and for ramming. So the same sensor kind of designs or chip designs are a bit of a platform, a bit of a workhorse. I mean, we can make multiple different types of electrodes. We have a variety of different materials in the fab that we can deposit and make sensors from them. And then obviously we can multiple sensors on the chips, so we can do multiplex analysis. And the reason we're using the fab and we're not, say, using drop sensor or any of these other technologies is because these are scalable for mass manufacture. So once we have a sensor design and material deposition that we want to use, we can then mass produce them using the microelectronics fabrication approaches, so we can make them in the millions. We then take those chips or sensors that we've made, and we've developed a variety of different types of electrochemical techniques that we use. More interestingly, in my group, we don't use 3 electrodes, we use 4 electrodes. So we do 2 working electrodes in parallel. So that opens up a generator collector techniques and a variety of other opportunities, which I'll go into briefly in a minute. We can obviously work across a wide variety of analytes. We do most of our analysis in liquids, but we also do some gas analysis as well. And I suppose finally, once we have all the once we have our sensors fabbed, to bring them out into the field, you need to have analog front ends to run them. Now you can buy commercial potentiostats, things like Palm Sense, but they cost €5,000 to buy. We build our own for a couple of hundred, and these homemade analog front ends, which I'll show you, are comparable, and we've benchmarked them against the Auto Labs and the CH Instruments, so the €25,000 laboratory equipment. So we can use all that. We can use these analog front ends, and we've written mobile phone apps now that will allow us to actually do the analysis in the field, use the phone as a gateway either to the cloud, or do some analytics on the phone itself. So I suppose if you look at that electrochemistry is very good in that it converts kind of chemical processes and reactions into a digital output. And that digital output, like I said, then we can use it to do analytics, we can use the calibrations, we can use to get qualitative data on what we're measuring. We can use a simple thing like our mobile phone to measure it. And one of the main advantages, I suppose, of electrochemistry is that it's cost effective, so it's reasonably cheap. It has good sensitivity. It has it gives you rapid results. You can get a digital output, like I mentioned. You can fabricate these sensors at high density, and your sample volume is very, very low. You can just 100 microliters of sample is more than enough to do measurements on. And when you go to the nanoscale, you don't deplete the analyte that you're trying to measure, so you actually end up doing very quick, rapid analysis, right? But so if electrochemistry is that brilliant, right? The question is, why aren't these sensors used everywhere? I suppose the most pervasive sensor is the glucometer. We're all familiar with that. But other than that kind of product, there's not a whole load of electrochemical sensors on the market. And the reason for that is that electrochemistry in general has a requirement that you use background electrolyte to block and shield charging. There's sample preparation involved. You need to maybe change the pH, or you need to pre-treat it depending on what you want to do. It's susceptible to interferent ions. So electrochemistry is brilliant at telling you how much electrons are there or how much of material is there, but it won't tell you what that material is. So if 2 species reduce or oxidize in the same voltage region, you're going to get false positives or false negatives, depending on what you're measuring. Like a lot of electrical electrodes, you get reference electrode drift. Like I mentioned already, the lack of specificity. So we need to develop materials to give us that specificity. And because there's individual kind of variances in a lot of the sensors that we fabricate, in a lot of cases, you need to do individual sensor calibration. So that actually slows everything down. And it's it's not really good for mass production. So to come around that, what we started working on about 10, 12 years ago was to develop very small volumes, where we could actually control the complete environment in this very small volume, and then really start understanding and digging in and exploring the interface between the electrode and the material of interest. So by doing that, then it'll ultimately allow you to develop very highly accurate and sensitive sensors that would have high reliability, repeatability, and reproducibility. So I mentioned my background is analytical chemistry, very used to using HPLC and these high-end analytical techniques, which are very highly reproducible and reliable. When we started doing electrochemistry using commercial screen printed electrodes, reproducibility was measured in hundreds of percent, not in the 0.1% that we would be liking. So that's the reason we dug in and we started doing silicon chip based electrochemical sensors. So here we have an example of one of our electrode designs. We have 8 different sensors on this chip. These are interdigitated electrodes, and so they're contacted from both sides. We've got a counter and a reference electrode here. And rather than using wire bond or flip chip technologies or anything like that to electrically connect to them, we designed the chips such that they have a pinout on the edge of them. So this is a HDMI. We also have SD. And they'll just literally connect in, and I'll show you later, they connect into a little a female HDMI connector on a PCB, and that's your sample or your chip electrically connected to your electronics. So it makes it really, really easy. We retain the metallization on the chip the same in more or less all our designs. And what we do then is the actual electrode itself, we change. So we can either make these using e-beam lithography for nanoscale, or a variety of different optical dimensions. So here you can see examples of the interdigitated electrodes at different gaps. So we have 1, 2, and 10 micron gaps between those electrodes. And because we can selectively bias each one of those interdigitated arrays independently, we can deposit down a variety of different materials just at those, just at whatever array we want. You can see example here, some silver ions after being deposited down for a chloride type of sensor. The nice thing is then that the other electrodes here, you can see them here, are not coated in any way. So we can use, so we have now 2 different types of electrochemical surfaces in a very, very small area. So that opens up different types of bimodal sensing, or it opens up generator collector, or it opens up pH control. And I'll go into that in a minute. So using these chips then, we're working on, my group focuses on 4 main areas in agri-food. So we develop on-farm disease diagnostics for animal health and welfare type of applications, looking at stress, looking at different types of diseases. We're really interested in food security, bacterial spores, residues, antibiotic or pesticide type of residues, allergens when you're moving in the processing stage, as you're moving from one product to the next. Is there a previous allergen you have to make sure is gone? We've done an awful lot of work on water quality, water sanitization, looking at chlorine concentrations, looking at pollution, looking at different types of contaminants, and plant pathology, where we're interested in diseases in plants as well at the growing stage. So a lot of these techniques would be would translate across the different types of applications areas that we're looking at. More recently, we're looking at soil chemistry. So we're interested in developing and have developed sensors for nitrates in soil, where we can measure nitrates for months on end with the sensors buried in the soil. But there's other applications in here like phosphates and nitrites and ammonium, just to close that whole nitrogen cycle. And then things like if you use an organic farmer, how much nutrients is in your manure, or how many nutrients are in your dirty water. So there's a key. This information is actually really important to farmers, because if they need to put nutrients or fertilizer on the soil, they need to know what they need. They need to know what's already in the soil, and they need to know how much therefore they have to add. So they need sensors effectively to tell them those data, to give them those data. So we developed a nanosensor platform, which I showed you 2 slides ago, and we measure now in a variety of different sample matrices, from the very, very simple water all the way up to very complex, such as milk. Milk is much more complex than blood or saliva in the sense that it has an awful lot of fat in it, and that fat can actually affect your measurements. So we've developed the sensors to cope with that. We measure, we do standard electrochemistry, for electrochemistry. So we have 2 working electrodes. We have the on-chip platinum reference and counter electrodes. And we can measure a lot of different kinds of ions and metals and analytes that you can typically measure in a lab. So anything you can do electrochemically in the lab using a beaker and a potentiostat, we can do on chip. We can then modify the electrodes to make them sensitive to different analytes. So, for example, from a chemical point of view, we can put on MAXenes, we can put on MOFs, we can put on MIPs, or we transition metal oxides, which will give us sensitivity to the electrode or to the material that we want. We collaborate with groups in Tyndall as well that do some DFT type of simulations for us that will inform us on what those materials should be. We also do a lot of bioelectrochemistry, where we can modify our electrodes with things like enzymes, with DNA, with antibodies, with proteins, with viruses, and check them for the complementary biomolecules in serum and milk. And finally, we can use the sensors to electrodeposit down different metals with nanospaced gaps between them, and we can use these for Raman type analysis. So if I just go through some of the work we do, the idea here is to get away from deep technical, but to kind of give you an example of some of the work and some of the facilities that are open to people through the INFRACHIP project that they can access from our group. So we started years ago doing COMSOL simulations to understand how electrodes should be designed, how they should be laid out to give us optimal sensitivity and selectivity. And when we did this, when we started, us and the rest of the literature were limited to 2D simulations because of the computer power that was required. 3-dimensional simulations were very difficult, and they kept crashing the computers. So effectively what we would do is we designed the architecture that we were trying to measure, and we would take a cross section through the electrode designs. Here is just an example of a single nanowire. And then we'll build a model that's 2D. So this little gold rectangle here is this 2D intersection across it. And we'd build the model and we'd look at different concentrations of different analytes, and the model would show back different results depending on the size of the electrode, say for here, or the concentration of the analyte. And then we could go away and we could change the design of the architecture and make the devices. But what we found was the results we were getting back didn't fit the simulations, right? So here's the 2D simulation. So this is the same problem statement, right? When we simulated the devices, we made the devices and we did the electrochemical measurements. And the blue data here is an example of the 2D simulations. And the black data were the experimental data. And you can see there's an offset between the 2Ds, which is quite high, right? So at best, the 2D simulations will give you kind of an order of magnitude estimation of what you'd expect. But as computer power improved and as our modeling skill increased, we were able to develop 3D simulations for the same problem statement. And we were also able to simulate the 3-dimensional models. And you can see that the 3D data, the red data here, sat on top of the experimental data very well. And that kind of that forced us to ask the question, well, what's going on and what are the problems? Because we knew the data we were getting back was quite good in terms of sensitivity, but we wanted to explain and understand this disparity. And what we realized was, I suppose the 2D simulations here, you got a calculation and then you integrated it along the length of, say, the nanowire. But what it completely omitted to understand was any diffusion effects at the edges. And as you can see, it's this edge diffusion here, either to or from the electrode, depending what the analysis is what results in this disparity. So that has changed how we now design some of our electrodes, and the architectures that we use is to try and understand the diffusion from the edges. And of course, what we've also done is measure then, well, at the small scale here, 50 micron scale, the length of the wire being 50 microns, those edge effects dominate. But when you move to much longer nanowires, at what point do these edge effects stop and the kind of radial diffusion profiles begin to dominate? So we've done a lot of work in that. Now we're applying it to different architectures and different types of structures, and that's given us similar data. So if someone has a need to try and understand their design, these simulation tools are available. What's interesting though is using interdigitated electrodes, we can bias these, like I mentioned a while ago, at different voltages. So we can use one, say the left one here, to do the sensing, but the right one we can use to electrolyze water. So if we put, if you remember, kind of chemistry 101 in secondary school, that if you put a current across two electrodes, you produce oxygen at the anode and hydrogen at the cathode. And as part of the production of oxygen at the anode, right? So when we're oxidizing this at 1.6 volts, we produce protons. And the opposite is true if you're reducing it, you produce hydroxyl groups. And you can actually, by producing these protons, you can tailor the pH in the area around the sensor. And so that allows us then to change the pH in a bulk solution from anywhere between 2, we can change it to between 2 and 12 without having to add in any acids or bases or buffers. So that opens the door up then for us to do electrochemical measurements at different pHs without the need to add in reagents. So for instance, if you're measuring in a pipe, or if you have a sensor buried in the soil and you need to change the pH, you can do it using this type of approach. And again, we use the simulation tools to understand, could this be done? So we simulated the production of protons. So at time 0, there's none. As you move on at time 1, you get the protons are produced and they start diffusing away from the electrode. And at 10 seconds, you can see there's a large diffusion. These are the optical micrographs then of these simulations where we use the methyl red pH indicator, which changes color from orange to red as it goes below pH 4.5. So you can see at time 0, it's all orange. And then as the time progresses, you can see the red production of protons effectively diffusing around the sensor. So we've changed now within this red zone, we've changed the pH environment. We've dropped it from what was initially pH 7 down to pH 4.3. And as the time moves on, you can see this, this red circle spreading out even further. So we can temporally and spatially change the pH around the sensors. So we can, in theory, and we've shown this, that we can have multiple different sensors measuring different things in the same sample, and each one of those sensors is measuring at different pHs. So that really allows us to do pH control. And now all of this work was enabled by the simulation tools to inform us on what the best type of design should be. So a challenge then for us is if you're changing the pH in such a very small area. So you can see the scale here, that's about 300 microns. If we change the pH at that scale, well then how do we know what the pH is? So a lot of people use iridium oxide and that for pH sensors. We didn't go down that route because it's a rare earth metal and it's really expensive. Instead, what we've done is we've used gold oxide as a proxy to tell us the pH. So here's a cyclic voltammogram of gold nanowire or microband in water, and you can see the black line, and we form gold oxide here. This is where we start producing molecular oxygen, but this is formation of gold oxide, and this peak here is the reduction of that gold oxide back to gold. And the location of this reduction peak is actually pH sensitive. So if we change the pH, we'll actually move and shift this peak. So the smaller the pH, the lower the pH, this peak shifts anodically. So we can use the position of that peak then as a proxy to tell us what the pH is. So by measuring the location and plotting it against pH, we get a nice straight linear line. And then literally by looking at the voltage of any peak, we can use this line to tell us what the pH is. So that works really, really, really well. And we've used that in a number of different sensing challenges now, and it's really, really good. And we've benchmarked it against the glass electrode, and it lines up pretty much perfectly well, even in real samples like coffee and milk and different types of water. So this is the actual using pH buffers to change the pH, and we can also do it electrochemically by changing the bias, which affects the driving current when we're producing protons. And so we can actually tailor that pH, like I mentioned a while ago, and we can measure the pH using the other interdigitated electrode, and we get a lovely, very good straight linear fit with an R squared there of 0.998. Now, the one thing we do notice is that the slope is 1 volt, not the 59 millivolts that we'd expect. And that's because we got supernormal state behavior. But that's okay, because the calibration takes that into account. Again, using these interdigitated arrays, looking at simulations, we can set up what we call redox cycling. So you'd bias one electrode at the oxidation potential, the other electrode at the reduction potential, and you get this cycling effect. So if we look at without using generator collector or the redox cycling, you deplete the analyte very, very quickly, and this is the black line there gives you the kind of response you get. But once you start reoxidizing the solution and setting up this cycling effect, you can see that our sensitivity increases many fold. So we can now do this reagent free, and we can now get really enhanced sensitivity just by changing the architecture, which again was informed by the simulation. So the simulation tools are very powerful and are very strong, and we can then superimpose other things on top of these, like electric field type of simulations or fluidic flow or hydrodynamic type of simulations. I'll show you the hydrodynamic stuff later on. So obviously we've made our devices now. So what's the next thing we want to do is electrochemically characterize them. So we have a variety of multiple different kind of potentiostats in the lab, which are available. So we have CH, we have autolabs, we have Faraday cages, and we have little portable PalmSense type of molecules as well, or types of molecules, instruments as well, which we can connect up to a phone, or we can connect to a laptop. So these would have been the first analog front ends or systems that we would have used when we wanted to go into the field. Number of different types of variety of techniques. We can do cyclic voltammetry on these chips, and like I said, these techniques are applicable to other people's sensors as well. If you want us to do some characterization, or if you want us to put some materials on it and understand how they work. Excuse me. So cyclic voltammetry is a really good technique. It gives us information on the different types of molecules that were there, where the voltages are that electrons hop on and hop off. And like I said, the redox peaks are pH sensitive. So we saw an example using CV a while ago to measure the pH. And we get some qualitative and a lot of quantitative information back from the electrode. We also have a different type of technique then, which is called square wave voltammetry. And this is the kind of, sorry, I should have said here, in the waveform here is you just sweep the voltage with time. So you sweep it up and you sweep it down, and this is the kind of response you get back. With square wave voltammetry, you're doing a different type of technique where you're putting on a staircase waveform. And you'd measure the voltage here, or sorry, the current here, and you'd measure the current here. And the idea of doing that is that it gives the sensor a chance to settle down in the environment, so it eliminates out any background charging currents. And then you can measure the forward sample, you measure the reverse current, and then you measure a delta, and it gives you this kind of peak shape. The nice thing about this is it's a lot more sensitive. It's very, very fast. It's a lot faster than cyclic voltammetry. We can change all different kind of parameters in there, like the frequency and the bandwidth, and that will give us information on kinetics as well. And it also reduces background charging, so we don't have to do any background calculations. And an example of where we've used this here is in silver ion detection in tap water, where we first electrodeposited any silver that was present in the solution onto one of our electrodes. We controlled the pH with the other interdigitated electrodes. We did, again here now we didn't have to add in any reagents, and then we stripped off the metal, right? And we used linear sweep square wave voltammetry, and we measured a different peak intensity for different concentrations of metal, and we were able to go down to quite low concentrations of silver. And this we benchmarked against using ICP mass spec. Now we've done this for multiple different metals. I'm just showing you silver here, but we've also used it for copper, we've used it for lead, we're using it for cadmium. So any type of metal is applicable here, and when you have multiple sensors, you can do it for multiple metals in parallel. I'll show this maybe a little bit more detail later on, but we use this technique as well to do nitrates measurement in soil. And we get very good sensitivity for our nitrates. Here, we've developed a material specifically, a metal oxide material specifically for nitrate. It's super selective to nitrate. We've done the background kind of calculations and measurements against different ions and counterions and anions. You see there's no real interference. The challenge we had here was that it worked at pH between pH 2 and 3. There's no soil, and well, there's no healthy soil in the world that's at pH 2 and 3. So to allow us to do measurements in the soil after we buried it, we had to use our pH control technique. So that's an example of the different types of techniques, and then the simulations informing our design, allowing us now to actually address a real world challenge, which is actually measuring nitrates in soil, for which there is pretty much no commercial solutions on the market at the moment. Then you can do a technique called electrochemical impedance spectroscopy. This is where you superimpose an AC signal on top of your DC waveform. And this gives us an example. This is particularly used, say, in the biospace, where we were measuring the layers of biomaterial on your electrode getting bigger as antibodies bind to their targets, or DNA binds to complementary strands. And this is a very sensitive technique. The challenge with it, though, is that if there's any small variation in the sensor fabrication, which there usually tends to be, well, then there's a complicated calibration kind of methodology required. But it does give very good mechanistic and kind of quantitative insights into the design that we're looking at, into the materials that we're looking for, and we can really understand the interfacial behaviors at that nano electrode molecular interface. So electrochemistry isn't particularly brilliant at telling you what something is. So to address that, we've developed a variety of different types of structures, which could be deposited on any type of electrode, really, not just our ones. So here's an example: some circular microelectrodes where you've used nanofoam, gold nanofoam, for really highly sensitive measurements of haptoglobin and cortisol. Again, the copper nanofoams can be used for the non-enzymatic based detection of glucose. We can use copper nanodendrites for herbicide type of detection. This is an example of Raman type spectroscopy. And then different types of materials. Here's one for nitrates. We can use phenylene diamine gold platinum for lactose and glucose type of measurements. And then we have different types of MOFs for PFAS type of measurements. So if you're looking at innovation in electrochemistry, it's really going to come from new types of materials and the integration of those materials onto state-of-the-art type of sensors. Sensor design would probably give you some incremental improvements, but it's the materials and the integration of those materials that'll give you the step change, the 10x type of improvement that we need going forward. We've also used the whole literature for gold chemistry uses thiols or sulfur chemistry to attach biomolecules to gold electrodes. We found those to be very, very unreliable, and you got very low reproducibility from sensor to sensor, let alone from device to device. So to come away from that, we've developed chitosan gold scaffolds that we electrodeposit down onto our sensors. Again, we can selectively here deposit it onto one comb and then use the other comb for pH control afterwards. And we find that these are conducting, so we get an increase in conductivity. So we're not creating any dielectric layer on top of the surface like you would with thiols. And then we can attach the antibodies or the DNA or whatever biomolecules you want specifically onto the chitosan. So that's working very well. Other types of materials, examples is, I mentioned there at the start, reference electrode drift is a problem. This is because the work function of the reference material, say platinum in this case, changes over time. So you get platinum hydrides, platinum hydroxides forming. And that changes, like I said, the work function, which means that if you're using that metal to pin the voltage on the solution that you're trying to measure and the work function changes, well, then your measurements change. So we put a lot of effort and time into stabilizing that reference electrode such that you end up moving from a situation like this to a situation here, which is much more stable, repeatable over hundreds of scans. And this reference electrode stabilization approach will work on any reference electrode. With regard then to antibody immobilization or any kind of biomolecule immobilization onto a chip, there's a number of different methodologies reported in the literature. So you have random covalent type of interactions, and then you have tried to do oriented interaction as well. So a key challenge with biomolecules is that if the antibodies aren't facing in the correct direction, well, then your reproducibility is going to be your lack of reproducibility is going to be very, very high. So we need to put a lot of effort into orienting antibodies if you're making bioassays or if you're making biosensors. So here's an example that we're looking for casein, where we use just random deposition techniques. We immobilize down the antibody using EDC NHS chemistry, but we're not controlling its orientation. And you can see from the results here, the differences. If you look at the delta, these are all the exact same experiment on different sensors. And if you look at the deltas between the different sensors for before and after mobilization, you can see that they're all completely different. And if you're trying to build a calibration plot on this, you'd be tearing your hair out because effectively it doesn't work. And you just won't get a reliable sensor. You like for a paper, if you used one, yeah, you get a great paper or PhD chapter. But if you're trying to make a device that you can bring into the marketplace or into the field, it doesn't work. So to come around that, we've had to look at different types of attachment strategies. And again, we're back to that controlling that electrode interface. And we've been using things like boronic acid now and protein G to actually orient the antibodies in one direction, which is facing up, so that the light chains here can bind to the antibody of interest. And if you look at the difference then between the random or the non-oriented immobilization, which I showed you a while ago, versus the oriented immobilization, you can see that the reproducibility here now is significantly higher, and the error bars are quite low. So this is very good, a very good approach for building different types of assays. And when you look at the differences between on-chip and between chip reproducibilities and different types of calibration plots, you can see that even here that the calibration plots using multiple different chips sit on top of each other. So this is important that we control that interface between the electrode and the molecule of interest. Okay. We've taken that technology then and we've brought it out into the field. So we've developed our own electronics, which I'll show you in a minute. Literally gone into the field. We're doing potato virus Y detection here. So this is where we're measuring a virus in plants directly. So we take a leaf, we crush it, we put it on the sensor. We were doing this in Ireland and it was summertime, so we bought a tent to keep the rain off us, and we kind of set up inside that tent. And what we found actually with this type of measurement, there wasn't lack of reproducibility that challenged us. It was the dust from the field was actually contributing into some of the measurements that we were measuring, so giving us a bit of a false positive. So once we cleaned up that and we eliminated the dust using a proper cartridge and fluidic cartridge, we were able to differentiate between infected plants here on the right and non-infected, on the left, and non-infected on the right, very, very clearly using the sensor. We benchmarked that against qPCR and ELISA measurements in the lab. So I showed you here, we did our own analog front end. So if I explain what we do here, typically electrochemistry, so this is simple enough. You have a reference working and counter electrode, and then what you need to do is to put electronics around that, where you have signal generators, amplifiers, different type of voltage meters, current measurements, and you're measuring in the cell, right? So to do that, we use a commercial analog devices chip, and that enables all the kind of electrochemical techniques that we want to do. And we've developed a process around where we were able to integrate these into a device. We start off first using a kind of where we use a large PCB that we buy from analog devices, and that allows us to do the initial type of design. So here you have the evaluation kit, and the chip is on it, and then you can program it using a variety of different approaches, and then put in a variety of different passives, so different types of capacitors and resistors, and understand the type of circuit that you want to do. And the approach to doing that is you'd start off with the you'd have the sensor input and the output signal characteristics you want. You design the electronics for that. You perform your tests in the evaluation kit. If they work, you design a PCB, which is much smaller, and then you go in and you do the whole debugging process. So I'm just looking at time now. I'm going to speed up a little bit. So you do the experiments in the lab using the commercial potentiostat. You'd go away and you use the eval kit and the programming to give you the exact same results, and then you would design the PCB. You'd fabricate the PCB, and then you'd characterize the PCB, and you'd expect the PCB to give you the exact same response as the autolab if it's working correctly. And you can see here an example of where we've done that. The black data is from the analog front end that we've done. The red data is from the autolab simple cyclic photomegram, and they work very nicely. And we also have an app now programmed on an Android phone that will actually allow you to do this in real time. So you can program the chip and read the data back from the chip via Bluetooth. And this is just a couple of different examples. And once you have the data on the phone, it saves it as a simple CSV file, and you can send it off to the cloud. So here's an example of if this video works. Maybe I have to get rid of this screen thing. So here's an example of some electronics. The AFE is in here. We've programmed it off camera, so we've already sent the test type of requirements to it. This is an example of one of our chips. We then put that chip into a microfluidic device or a fluidic device that we 3D print, and I'll explain that in a minute. And the 3D printed device then, and this will work for most different types of sensors as well. So that just connects in using the SD pin out to the electronics, and then puts in the sample. Sample is just dropped on here, just a few hundred microliters, and then we do the measurements with the mobile phone, and you can see how fast the measurements come back. So this is done through Bluetooth. The data is read by the analog devices chip in here, and then a simple Bluetooth 5 device sends it to the phone. So that's an example of where you want to go into the lab, or where you want to go into the field, and you want to have low-cost electronics. That's the type, or if you even want to bury the electronics in the field. That's some of the problem statements we're addressing. Microfluidics, we've done a lot of work on this. We've used every different type of techniques from PDMS and REE bonding to the chips to xerography, where we've used pressure sensitive adhesive. And we didn't have much success. Like, we got some results, but there was a lot of leaking and problems. So you make 10 devices and one might work, right? So it's a very slow approach. So what we've done instead is we come up with a resin 3D printing approach where we can use multiple different resins. We design them in CAD, and then we 3D print the resins. And we use different, like, flexible resin to form a gasket or a seal to the chip, and then a harder resin on top of it to make sure it's robust and it'll survive the environment. And we've done, again, we're back to simulations to understand how hydrodynamics affects the diffusion, and particularly around pH control on a chip. So we've done those measurements, and we can see that because it's laminar, the pH control still works on the flow. So we use the microfluidics. Here's an example of it, electrically connected again, and this is food dye that we just injected across, so we could actually visualize that the fluidics were lining across the sensors. And we've done measurements under static with no flow, and then once we put on a flow, and you can see that we get another massive, huge enhancement in sensitivity there. This is just a video of fluidics. I won't show it because we're running out of time. So the next thing then is packaging, right? So if you're putting a sensor into a field, you need to package it up. And there is no solution to what we wanted to do. So again, we're back to 3D printing. We're back to protecting the sensor using hydroscopic or hydrogels. So we synthesize hydrogels, we place them on top of a sensor within a 3D printed cell that we can then electrically connect to using the pinouts, and then we passivate all the connection as well. It's not shown in the photograph here, and we characterize and benchmark it so we can show that the hydrogel works, that the packaging works, and then we can actually deploy them into the field. And then to protect the wiring itself, we have an outer packaging housing that goes over the chip, and this passivates all the electronics so no water gets into it. And using that approach, we've actually developed nitrate sensors. Here's a picture again where we've dug a hole in a farm, and we've measured them. We started off by doing flower pot type of measurements in the lab, and then we went into the field, and we got very good data from that. So finally, as well, I mentioned earlier about calibration and the challenges around calibration and the slight variations in fabrication give you. So one of the approaches that we're doing now to address that is to use different machine learning tools to build up a model of all the different types of calibration plots, or sorry, not calibration, CVs or square waves that we get, and then using either spike neural networks or, in this case, principal component analysis to build a calibration model. So this example here, it's for pH measurements, so identifying where that reduction peak is. This is the same CV I showed you a while ago, but it's split now. So instead of it going back this way, it's just still going forward. And we were able to build a model where we measured different pHs from 3 all the way up to 10 at a variety of different temperatures, so from 4 degrees all the way up to 80 degrees, and use all those data to build that model. So when Lingy was doing this, he used 900 data sets for the model, and then he tested it against nearly 450 other sets. And he used 32 principal component analysis, and he got a really, really good model where we could measure the pH of a solution regardless of the temperature, and it gave us the pH of that solution with 95% accuracy. So some sample solutions or sample kind of application areas using pH control. We're interested in measuring chlorine in tap water. Why do you need pH control? Well, tap water, depending on where you are in the world, can be anything between maybe 6.5 up to 9. So if you're trying to measure that electrochemically, you get completely different measurements. So to do that, you have to add colorimetric and buffered chemicals to the solution. Here's an example. If you just do it at pH 8 electrochemically, you get this type of sample. If you acidify it, you get a much higher sensitive measurement, which is what you want to do. So if you acidify this, you're driving everything down into this regime, and you're measuring the gray here, the hydroxychloride. And using pH control, you can see that the pH acidified with acids gives us the exact same data. So we're able to measure chlorine now without the need for any selective membranes or any added materials to the devices. An example, I showed you silver a while ago. This is us out on the field, Rob measuring copper next to a copper mine, and again getting very high sensitivity for copper. And benchmarking our measurements against the ICP mass spec. So they correlate very, very well. We can immobilize enzymes onto the substrate. We can get measurements for glucose, lactate. What's really interesting, the glucose is glucose oxidase optimal activity is a pH 5.5. Different, we're measuring here calf saliva. And the pH of the saliva itself changes dependent on the health status of the animal. So if you're looking here at the ranges, you can see the pH ranges of these different calves. It goes from 8.2 down to 5.3. Well, then the pH measurements you're going to get back, or sorry, the glucose measurements you're going to get back are completely wrong. So you have to do pH control on the saliva, and you can then discriminate very, very quickly what that glucose concentration is. And you can see that these first 4 calves are healthy, whereas this last calf here is quite sick. And the data comes back and gives us those values. The pH on its own wouldn't tell you that, but we needed the glucose measurements as well. This is some of the data I showed you a while ago. This is interesting. This is nitrate measurement without pH control. This is nitrate measurement with pH control. This allowed us to put the sensor in the field. We flew the field with a drone. This was an EU project called Sarmenti, led by CEA in France. Where we were developing the sensor element, and there was a back-end server, there was an SDL data logger, there was LoRa communications to actually send the data from the soil back to the farmhouse. There's the LoRa mast on the farm. And to do this project properly, we flew the farm, we got some fluorescence images. The granularity of those fluorescence images was 2 microns. And so you can see he was able to map the 23 hectare farm here quite well. We buried our sensor in this location. And when you went in, you saw different colors of green and yellow. And the question was, what does green and yellow mean in terms of concentration of nitrates? So they were using the chlorophyll in the maize as an indicator. So to do that, they took the nitrate concentration from our sensor, and then we were able to attribute that to the other colors. We also, during that project, benchmarked the measurements by taking soil samples and sending them to the lab. And the lines here are just guides to the eye. The data points here are really interesting because you could see that these data points are taken about 10 days apart. And between this one and this one, it missed this whole fertilization approach where the farmer putting that nitrate onto the soil and an irrigation here. You can see the nitrate got really high and then its bacterial activity here kicked in. The nitrate concentration went high. But the lab measurements here and here missed that completely. So it's temporarily not very good. And like this is taking a soil sample every 10 days is brilliant because normally it's done about once every 3 years. But you can see our sensors were given measurements back twice a day. And we were able to really understand the dynamics of the nitrogen in the soil when we were doing that. And based on that map, then the farmer was able to program an old type of fact using just GPS. It was an old tractor. It wasn't a really fancy one. And it developed a whole soil strategy, fertilization strategy for it. Just basically by speeding up and slowing down the tractor, he could change the concentration of nitrate he was putting on the soil. And what worked for him was he used 500 kilograms less of the fertilizer over the field, which probably saved him about €500. But more importantly, it really reduced his environmental risk because one of the very first slides I showed was the nitrates directive by the European Commission. And that directive states that the polluter pays, right? So if any nitrates gets into a river and there's a fish kill or there's any damage, the farmer is liable to pay the costs of whatever happens as a result of the runoff. So by knowing exactly how much nitrogen you should put in the soil and how much you need, he was able to use a lot less fertilizer, which reduced that risk of him contaminating the water courses. And also then you can correlate your fertilization with weather. So that's a snapshot. I think I kind of kept the time, maybe a bit slightly over. That's a snapshot of where we are. We've developed these sensors. We can help people develop sensors. We can use simulation. So what's on offer to INFRACHIP, I suppose, really? We can do the sensor simulation and design. We can do not the electrothermal, it should be electrochemical characterization. We can do a variety of different types of surface chemistries, develop electronics for sensors, develop fluidics for sensors, and also help to develop machine learning. So that's me finished. So I'm going to stop sharing now and maybe go for some questions and answers.
All right. Thank you very much. This was a very interesting presentation with a good number of practical examples. So again, I remind our audience that we have the question and answer tab where you can write your questions. And it looks like the first question we already have here. I would like to know why you're using 4 electrodes, including 2 working electrodes, and what exactly these were utilized to study.
Yeah, so like I probably showed after that question came in, so we could use them to control the pH, right? So had applications in a variety of different types of analysis, from heavy metal detection to nitrates detection to glucose measurements. We could also set up then a redox cycling where we were able to cycle the molecule and get enhanced sensitivity. And which I didn't mention in here, you could actually take out background interference effects as well. So if 2 molecules oxidize at the same place but reduce at a different place, we can oxidize the 2 of them in one position and we can measure the oxidized species at a different voltage. So that allows us, say, to do dissolved oxygen measurements where we convert the oxygen to hydrogen peroxide, and then we detect that hydrogen peroxide, and that removes effects from iron and chloride and all other kinds of things in the analyte. So it's all for us. We try to increase the sustainability of our measurements by not using additional reagents, and also that cuts down on any possible contamination or dilution effects by adding these reagents can cause.
All right, thank you. And it looks like Daniela has her hand up. You can go ahead and open your microphone and ask the question if you want. Okay, I guess this was an accidental hand.
Okay. The next question, how did you power the sensors? Do you also develop self-powered sensor using energy harvesting technique? How much is the minimum power for powering these sensors in farming application?
Yeah, so really good questions there. Yes, to all of them. So normally in the handheld type of prototypes that we have, we use just a simple mobile phone battery, right? And they last for months. You don't have to charge them very often. So it's the electronics as opposed to the sensors that draws most of the energy. So you're looking at a kind of couple of milliwatts. With regard to the soil measurements within that project, we did have a PV cell and an external battery measuring the whole system, but there was multiple other sensors, including gas sensors, involved in that, so that it was quite power hungry. But these sensors, we haven't done energy harvesting like on-chip vibration or anything like that. But we have used PV, but the applications we're using, the simple mobile phone battery, will keep it running for months.
All right, thank you. Do we have any other questions? Okay. The next question is, what environmental factors were considered during testing using this commercial chip that you mentioned, and how did they affect the system's performance?
Yeah, so we developed that on the bench in the lab, so we weren't able to really control the environmental factors in terms of temperature. But analog devices already had that done as a product, so it's a product, right? So what we did was everything we did, we benchmarked it against, or everything we do anyway, we benchmark against state of the art. So when we were doing the ADI chip, we benchmarked it against the Autolab potentiostat. So we do measurements with the circuitry that we had designed, and we benchmarked that against the Autolab, and you could see those data that I showed that they sat exactly on top of each other. So the environmental factors we didn't have to go into because we weren't developing the chip itself, right? So because we're doing electrochemical sensing in water, we're not affected by humidity, or we're not really affected by pressure. So it was just can we get that to give us the same measurement as the Autolab? And the answer was we could. And then we were able to, we also benchmarked it against the PalmSense, which ironically, right, has the exact same chip in it. So the PalmSense, the ADU 355 is the chip that runs that PalmSense. So we were reusing the same chip, so we were able to compare oranges and oranges. So we were able to do a direct comparison.
Could you mention, so again, the first part of the question was about the environmental factors. So I don't know if you've done things like changing the temperature or some other environmental parameters.
Yeah, no, it's relatively stable. I mean, when you're going into the field, like we're not doing automotive work here where you're going from -40 up to a couple of 100 degrees, and the temperature variation is within maybe 20 degrees. So when we were doing measurements, we obviously, whatever temperatures were in the lab is what we used. But what we should do ideally, and we've put in an infrastructure proposal to Research Ireland to get funding for this, is you should have growth chambers, right, where you can actually change a bit like a reliability temperature in microelectronics, where you can actually control very accurately the temperature, the light, everything, so you could replicate environmental factors. Because the challenge is, in the sensors that people are developing now, if you're putting something in the field, depending on whether it's summer or winter, you do affect temperature, right? And you need to address that. When you're working in the lab, the labs are environmentally controlled, like 18, 20 degrees plus or minus 1 or 2. You could use ovens, but they're clunky. Whereas if you have these proper growth chambers, you can control a lot more effects, and you can do multiple sensors in parallel as well. So it's work that we have envisaged, but we need some infrastructure to allow us to do that. We worked out about €3.5 million worth of infrastructure to allow us to do it properly. So hence we're trying to get funding for it. But it's a really good question, not just on the ADI chip, but on the sensors themselves. How does different environmental impacts change the results you expect to get? When we were buried in the soil, we were 20 centimeters down, so the temperature was probably about 10 degrees and didn't change much. But if you were in a barn, it could be anything from -10 up to maybe 40. So we have to do those measurements, but again, you'd be challenged by the equipment to allow you to do it right.
Okay, thank you. Do we have any other questions? Let's give it a little bit more time. Well, while people are thinking about maybe the last question, I'm curious if for some of the metals that you're using, you have experienced some corrosion or delamination or those kinds of
not really, no, not with the gold. The gold and the platinum are fairly stable, right? They're very good. The platinum will change a little bit, but when we put that stabilization layers on top of it, that kind of controlled the work function of that. Gold is quite good, which is why we use it. We don't like if you put silver in there, yeah, it'll kill you. We haven't really used carbon-based electrodes because we can't deposit them in the fab. And we've put some reduced graphene oxide and stuff onto the electrodes, but that's not highly reproducible. We are interested in boron doped diamonds, so we're developing those at the moment. So we'll see how they affect with time, but they should be fairly stable as well. But the gold and the platinum are good enough for us.
I think I was particularly curious about, for example, the electrodes that you use for pH control, because I guess you consume some of those, right?
You can, yeah, but you just make them thicker, right? So they're consumable anyway, right? But if you want them to last longer, you can either have multiples on the chip, which we multiplex them, or you can just make thicker gold. So that like you do delaminate, like when you're doing CV, you do ablate some of the gold away, and eventually you end up with none. But you can figure out when that's happening, and then you can swap to another electrode.
Okay. Thank you very much. All right. So it looks like we're not getting more questions from the audience. So I thank everybody for joining this webinar. Again, I remind you that this webinar series is brought to you by the INFRACHIP project. And as you heard a few times during the presentation, access to these platforms and technologies is available through INFRACHIP. So if you saw something that you think may be interesting or useful for your own research, definitely get in touch with the INFRACHIP project, and we will make the connection, and we will see what we can do for you. And with this, we can close. I remind you that we have one more seminar, one more webinar in the series, same day of the week, same time next week. And it looks like we do not have any more questions. So with this, I thank the speaker again, and I thank you for attending this webinar.
Okay. Thank you very much. All right, cheerio.